Welcome to episode 3 of Dr. Snow's Journal Club, where we review the hottest papers in brain health from the top medical journals in the world. Let me once again introduce you to world-renowned neuropathologist, inventor on 350 plus issued patents, and entrepreneur, Dr. Alan Snow, who will guide us in today's Journal Club. Dr. Snow, what is the paper that we're going to review today?
Hi, Scott. Welcome to episode 3 of Dr. Snow's Journal Club. Today, we're going to mainly review a paper that was published in this prestigious journal, Nature. So it's one of the top 2. In February 2024 by Murdoch et al. And it was entitled Multisensory Gamma Stimulation Promotes Glymphatic Clearance of Amyloid. So this paper has to do with the discovery in the last few years that light and sound therapy at a certain wavelength, believe it or not, specifically at 40 Hertz, actually stimulates the removal of amyloid plaques from the brain, and this has been studied in robust mouse models of Alzheimer's disease. This renovating discovery took 2 years to publish in the second leading journal in the world, with the number 1 journal being Science and the number 2 journal in the world being Nature. I believe that this paper took so long to publish because it was controversial. And my experience is that in controversial papers, especially novel discoveries, it involves convincing 3 reviewers that this paper is worth publishing in the major medical journal like Nature. I personally published 2 papers in Nature Scientific Reports, which is an offset of Nature, in 2019 and 2021, and both those papers went through a bunch of revisions to be published, and in accordance with the reviewers' suggestions, it took up to a year for me to get each of those published since I submitted them. So it's a lot of work.
Sounds like it's a lot of work and a lot involved with the submission. So, Dr. Snow, before this paper was published, there was a major paper in 2016 that led up to these discoveries. Let's first review this 2016 paper that discusses light therapy for the treatment of Alzheimer's disease. Can you talk about this paper that was published in the Nature Medical Journal? What was this paper about?
Yes, in 2016, a paper was also published by Lecarino et al. In Nature, and it was entitled Gamma Frequency Entrainment Attenuates Amyloid Load and Modifies Microglia. Quite fascinating. Here they found that light therapy at a specific wavelength of gamma waves, which is at 40 Hertz, but not at other wavelengths, reduced brain levels of beta amyloid protein. Remember, this is the protein involved in the formation of amyloid plaques. And remember, we talked about brain amyloid plaques before on the Dr. Snow Brain Health Show, and we referred to these as these large Swedish meatballs that accumulate in the brain in between neurons when you start aging, starting in your late 20s. And it tends to be neurotoxic, and it mucks up the connections between neurons to remember thoughts. So they play a role in decreasing memory and cognition as they accumulate. So get this, activation of local circuits of excitatory and fast spiking inhibitory neurons that resonate at 20 to 50 Hertz give rise to oscillations in the local field potential, LFP, called gamma oscillations. And in the 2016 study, the researchers recorded neural activity in a well-established mouse model of Alzheimer's disease known as the 5X FAD mouse model, familial Alzheimer's disease mouse model. And what they found is that gamma power was significantly lower in the brain of Alzheimer's disease 5X FAD mice than in wild-type non-Alzheimer's mice. So these results revealed that deficits in gamma modulation of spiking in a mouse model of Alzheimer's disease prior to the development of major amyloid plaque accumulation or evidence of cognitive impairment. And these gamma frequency deficits have also been found to be alters in humans with Alzheimer's disease. So get this, using light pulsation at a wavelength of 40 hertz, but not at other wavelengths. They found that 1 hour of light stimulation, so they're, beeping the light at different frequencies, and at 40 hertz, it reduced beta amyloid protein 1 to 40 in the brain by 53.22%, and beta amyloid protein 1 to 42 by 44.6% in the 40 hertz treatment group. So these are mice that are getting light blinked at a certain wavelength. And remember, these are the two main isoforms of beta amyloid protein in the brain of Alzheimer's disease, the beta amyloid protein 1 to 40 and the beta amyloid protein 1 to 42.
Huh. That's really interesting. So what did the 40 hertz light therapy do to the genes in the brains of these transgenic mice?
So get this, interestingly, 35% of all upregulated genes had their highest expression in microglia in the brain. Remember, the microglia are the Pac-Man in the brain. They clear out all the garbage and junk in the brain, like a Pac-Man. And the RNA sequence analysis strongly suggested that the 40 hertz stimulation caused an alteration in the state of the microglia, which is significant given the accumulating evidence that microglia play a role in Alzheimer's disease pathology. They also observed almost twice as many microglia in the 40 hertz group compared to other frequencies or random control groups. So the microglia also got bigger, and they showed increased evidence to engulf and eat the beta amyloid accumulation prior to plaque accumulation. Thus, the light therapy at 40 hertz was making the microglia engulf more beta amyloid protein, showing co-localization of beta amyloid protein within the microglia cell body to about 85% following 40 hertz stimulation. So it went from 31.7% to 41% following the 40 hertz stimulation.
So what did they find with light flickering at different frequencies with Alzheimer's transgenic mice?
So 3-month-old 5X FAD transgenic mice that hadn't accumulated amyloid plaques in the brain because they're still too young. Get this, they're placed in a dark box and they're exposed to either light flicker at different frequencies. So they looked at 20, 40, or 80 hertz, random flickers, or constant light, or in the dark for 1 hour. And 1 hour after 40 hertz flickering, beta amyloid protein levels in primary visual cortex was reduced by 58% compared to the dark or the light controls. Now get this, the effect was specific to 40 hertz flicker, as neither constant light nor 20 hertz or 80 hertz or random flicker significantly reduced beta amyloid protein levels compared to dark and light controls. So it's really specific.
That is very specific. So what did this 40 hertz light flickering do to the transgenic mice that were older and already accumulated amyloid plaques in the brain?
Yeah, that's a good question. So they next treated 6-month-old 5X FAD mice, which have established brain amyloid plaque pathology in many brain regions, and they used it for 1 hour daily over 7 days with 40 hertz flicker or dark, and then they analyzed the brain tissue, the visual cortex, 24 hours later. So get this, the 40 hertz flicker significantly reduced plaque number by 67% compared to being in the dark, which are controls, and the plaque size was reduced by 64% due to this 40 hertz flicker. So taken together, these experiments have now identified a non-invasive treatment with a profound effect on amyloid plaque pathology.
Huh. So did they ever look at the effects of this 40 hertz light flickering on tau pathology and tangles in the brain?
Yeah, that's a great question, Scott. Now you're thinking like a neuroscientist. They next used a tau P301S tauopathy mouse model. And they found that 7 days of 1 hour, 40 hertz visual flicker reduced phosphorylated tau protein in the brain by about 42%.
Wow. So, what were the conclusions from this study?
So the robust reduction of total amyloid levels in the brain was likely mediated by both decreased amyloid plaques in the brain and increased endocytosis of amyloid plaques and beta amyloid protein by microglia. Thus, it appears that driving 40 Hz gamma oscillations may induce an overall neuroprotective response that recruits both neurons and microglia. Amazing.
It is amazing. So I also know you mentioned the paper with Murdoch and company following the theme that 40 Hertz gamma light stimulation can promote clearance of amyloid plaques in the brain. So what is the premise of this paper published in 2024, also in the prestigious journal Nature?
Yeah, so that's the paper that we're reviewing today. So it's known that glymphatic movements of fluid through the brain removes metabolic waste. And you remember we talked about this, that sleep is actually good for you because when you have restful sleep, there's a glymphatic system that actually clears out the amyloid plaques and the junk in the brain. So that's why it's always good to get good sleep. Remember the snow brain principles, the snow brain, the S on snow brain stands for sleep. So it's very good to get restful sleep. So in this 2024 paper, the researchers showed, and by the way, the primary group is from MIT in Cambridge, Massachusetts, and they show that multisensory gamma stimulation promotes the influx of cerebrospinal fluid and the efflux of interstitial fluid in the cortex of these 5X FED mouse model of Alzheimer's disease. So this paper, published in 2024, about 10 months ago, tries to understand further how this 40 Hertz light can influence the clearance of beta amyloid protein and amyloid plaques from the brain.
Great. So let's discuss a little bit about the glymphatic system, which appears to be the basis of the paper. What is the glymphatic system and what does it do?
So the glymphatic system is thought to have a critical role in clearing metabolic waste from the brain. So like I said, when you get restful sleep, sleep is actually good for you as it helps the glymphatic system clear out metabolites, including pathogenic proteins such as amyloid plaques. So aging, Alzheimer's disease, and even the lack of sleep will impair the fitness of the glymphatic clearance of beta amyloid protein and plaques, as well as tau protein and tangles. So therapeutic interventions to promote glymphatic clearance have been hypothesized to alleviate Alzheimer's disease pathology. So this group and others now have found that non-invasive 40 Hertz sensory light, and actually sound as well, stimulation reduces amyloid burden in Alzheimer's disease mouse models. So it's not only flickering light at 40 Hertz, but also sound stimulation at 40 Hertz.
Okay, so we have the light flickering, and then we have the sound stimulation also at 40 Hertz. What does 40 Hertz sound like?
I'm going to put it on our video right here, and I started looking across YouTube, and I found it, and so let's play it now for a few seconds, and I'm going to put it on this video. So let's listen to what a 40 Hertz sound sounds like. By the way, it's a very low tone. Sounds like a space alien is about to land on Earth. It's very calming and peaceful to your brain. Also, for comparisons, the fundamental frequency of a male voice is 90 to 155 Hertz. I looked it up. and a typical female voice is 160 to 255 hertz. So this is definitely on the lower tone of frequencies. It's effective for improving, get this, concentration and focus, and it's associated with gamma brain waves, which are associated with higher levels of consciousness and alertness.
So it sounds like an alien spaceship. Little creepy, but soothing. But this is amazing to see what 40 hertz, whether it's light or sound, can really do.
Yeah, Scott, it's like in the Alien Takeover of the Earth movies, when there's a subtle, creepy, low-tone background sound, like they're trying to talk to us.
So, moving on with this paper, what did these researchers find that gamma stimulation does to the glymphatic clearance?
So these researchers also reiterated the fact that 40 hertz multisensory audio-visual stimulation increased 40 hertz local potential power in the frontal cortex areas of these 6-month 5x FAD transgenic mice, as expected on the basis of prior reports in mice and humans. So the 40 hertz stimulation attenuated amyloid burden compared with no stimulation, compared to 8 hertz or even 80 hertz stimulation controls. And using tracers to test whether glymphatic clearance is associated with 40 hertz mediated amyloid clearance, they then monitored the cerebrospinal fluid dynamics using a fluorescent tracer. And what they found is increased CSF accumulation in the cortex following 1 hour of multisensory 40 hertz multisensory stimulation. So these findings indicate that amyloid is cleared from the brain following 40 hertz stimulation, in part via the glymphatic routes. They then found that the multisensory 40 hertz stimulation increased arterial pulsations compared to non-40 hertz control stimulations. They also found that multisensory 40 hertz stimulation increased meningeal lymphatic vessel diameter. So the lymphatic vessels are actually changing compared to control stimulation in 6-month-old 5x familial AD transgenic mice. Great, amazing stuff.
That is. So did they look at the genes that were turned on and off by this stimulation of the 40 hertz?
Yeah, great question, Scott. So get this, they found that multisensory gamma stimulation resulted in 144 downregulated genes and 219 upregulated genes, which included astrocyte and astrocyte feet that played a role in the stimulation as well.
These are really amazing studies. So what conclusions were drawn from this study?
So in this study, given that the glymphatic system clears parenchymal amyloid plaques and it's regulated by neural rhythms, they hypothesized that glymphatic clearance might account for amyloid reduction by sensory gamma stimulation. So gamma rhythms may be relevant to aspects of glymphatic clearance, as gamma may be involved in aspects of sleep and neuronal synchrony. So glymphatic influx is also related to sleeping state. A recent report in 2023 suggests that non-invasive sensory stimulation modulates cerebrospinal fluid outflow in awake humans, potentially suggesting that cerebrospinal fluid and glymphatic pathways may be modulated in awake humans, suggesting that CSF and glymphatic pathways can be modulated in the awake brain. So these findings suggest that multisensory 40 hertz stimulation promotes amyloid clearance from the brain and activates glymphatic pathways. These results also suggest that vasoactive signals from neurons act on glial and vascular cells during neural oscillations to drive cerebrospinal fluid clearance. So it's a lot of interconnection between neurons, astrocytes, microglia, and clearing out the amyloid.
These are such neat concepts to study, and this is a neat paper to spend some time digesting with our audience. So could we hear the 40 Hertz sound once again to close the third episode of Dr. Snow's Journal Club?
Yeah, sure thing, Scott. I'm going to play these alien 40 Hertz sounds and lights. Here it is again, the 40 Hertz sound that will increase your focus, concentration, and memory. And then we're going to add a 40 Hertz flicker, and you will feel something different in your mind. Until next time, for another hot paper to review on Dr. Snow's Journal Club. Thanks a lot, Scott.
Thank you, Dr. Snow.